Okay. So yeah, thank you so much. And thank you to all the organizers for this great opportunity to present my work. I'm really excited to be here and talk to you all about today how single-nucleus multi-omics identifies insulin-responsive variant effects in skeletal muscle. So my name is Ben. I'm a graduate student in bioinformatics in Steve Parker's lab at the University of Michigan. And let's just dive into it. So this is a brief outline of what I'll talk about. I'll give a short background and motivation, and then I'll talk about the experimental design. I'll show you how chromatin accessibility and gene expression respond to insulin stimulation. And then I'll try to spend a bulk of the time on EQTL, CAQTL, response EQTL, and how that all ties together into T2D GWAS signals. I'll talk about primed enhancers and then end with a few conclusions and future directions. So our lab focuses a lot on type 2 diabetes, or T2D, and T2D is really this multi-organ disease characterized by chronic hyperglycemia. In T2D, insulin is a principal stimulus, so it's a hormone that stimulates glucose uptake by peripheral muscles, specifically skeletal muscle, adipose tissue. In this project, we'll mainly be focused on skeletal muscle. And T2DGOS have been enormously successful to date. So this is the most recent 2024 GGI project, which identified more than 1,200 risk signals across the genome. But deciphering the molecular mechanisms for these risk signals has remained pretty difficult. And there are a lot of reasons for that. One being that many of these risk signals, approximately 90%, fall in the non-coding portion of the genome. And another reason is that a lot of these risk signals may exert influence over a disease state only in specific cell types. So perhaps whole blood, or for type 2 diabetes, we commonly think of pancreatic islets, skeletal muscle, adipose tissue. And furthermore, a lot of these risk signals may exert influence on the disease only in specific stimulatory contexts. So we can think of disease-relevant stimulatory contexts. And a central hypothesis for this project, and what I'll talk about today, is that a greater understanding of both the cell type specific and stimulatory context specific regulation of these risk signals can help us better understand T2D, other polygenic diseases, and potentially identify future druggable targets. So in our project, we're using a technique called the insulin clamp. And in an insulin clamp, insulin, which is a very powerful hormone, is infused into the body at a constant rate. And insulin then stimulates glucose usage, principally by skeletal muscle in the body. And then to match the body's glucose usage, an equivalent infusion of glucose is constantly infused into the body to replace the usage over time. In our experimental design, we included 47 individuals, all of whom had self-reported Hispanic ancestry, and approximately half of whom were female. All of these individuals received a pre-clamp biopsy in the vastus lateralis. This is a skeletal muscle in the quadriceps. And this was before the insulin clamp. So then they underwent the 4-hour insulin clamp. This is that stimulation. And then they all received a paired biopsy, again in the vastus lateralis muscle. And this is going to be called the post-clamp sample. So 47 individuals, 94 paired biopsies. The samples were all then frozen and then shipped to the University of Michigan, where they were processed in 6 randomized batches with genetic multiplexing. And then they were sequenced with multi-omics, so single-nucleus RNA and single-nucleus ATAC-seq on separate nuclei. We then jointly clustered the data, but again, just to reiterate, we had 94 paired samples here, and after rigorous QC, we identified approximately 157,000 total nuclei. And when we jointly clustered those RNA and ATAC nuclei, 15 distinct clusters emerged. And we identified really these canonical muscle cell types, which agrees with previous results. So type 1 fibers, these are slow twitch muscles used for like walking or endurance activities. Type 2A, or intermediate fibers, and type 2X, or fast twitch muscle fibers, which are commonly used in sprinting or weightlifting activities. And these were the 3 most common cell clusters. We also identified FAPs. This was the fourth most abundant, and these are fibroadipogenic progenitor cells. These are multipotent stromal cells, as well as endothelial cells and 10 other unique clusters. Today, I'll really focus on these top 5 most abundant clusters, but we're running all analyses for all cell types. So this I'm going to bring into the slide. This is a multi-layer omics diagram, and we can see the genome there on the bottom and how genetic risk may be encoded there and then percolates through the different layers of the epigenome, the chromatin accessibility, the transcriptome, gene expression, and so forth until we get to physiology and disease risk. And really the question that I'll use to tie together a lot of these analyses is how do these omics layers change in response to this very powerful insulin stimulation that is also disease specific to type 2D or T2D. And the first analysis that I'll show here is a PCA dimension reduction, and we're taking each sample's single-nucleus attack-seq data, so all the chromatin accessibility data, and trying to condense that all into one point on this PCA plot. And canonically in PCA, PC1 is the factor that drives the most variance in the data. So the single most important factor that explains variance in the data, and the y-axis is PC2, or the second most important factor that drives variance in the data. And you can see here immediately there are really two clusters that initially emerge, and these are preeclamped female and preeclamped male samples. And we see that that's oriented along the x-axis. So biological sex here is really a separating factor in the data, which reflects this known sexual dimorphism in skeletal muscle. We also see here now the post-clamp female samples. So these samples are separated in PCA space based on the stimulatory context. And if we connect the lines that connect the samples from each individual, we can see that they're oriented along the y-axis. So the stimulatory context is really the second most important factor that drives variance in this attack-seq data. And similar concept for the male samples. So four relatively distinct clusters are emerging here in the attack-seq data. And when we do the same analysis for the RNA-seq data, again, we have four clusters, but now the lines you can appreciate are oriented along the x-axis. And here, the stimulatory context effect, that clamp effect, that new effect is now actually the most important driver of variance, is the most important factor in the data that can explain the variance. Outweighing all other factors, including biological sex, which you can see here is oriented along the y-axis. So just to kind of put them side by side, on the left, we see the chromatin accessibility, where the most important driver of variance is biological sex, and that outweighs the stimulatory clamp effect. But actually in the transcriptome, the RNA-seq, we see that there's a very strong, very robust effect that is being represented by that new stimulatory clamp. And this can really set the stage for, okay, well, if this transcriptome is really changing robustly, what does that mean for how genes are differentially expressed between conditions, and how does the genetic regulation for these genes change between the basal and stimulatory contexts? Returning to this, this multi-omics layers diagram, we can see and appreciate that the genome is essentially invariant across this 4-hour insulin stimulation. The chromatin accessibility is changing to a certain degree, but in the transcriptome, again, that stimulatory clamp effect is outweighing all other drivers of variance in the data and is really a very strong and very robust effect over those 4 hours of stimulation. So this is the differential expression results. So we see thousands of significantly differentially expressed genes in all of these 5 clusters, progressively decreasing with decreasing power. So we have the most power here in the type 1 fibers, those skeletal muscle fibers. And we can appreciate that almost a third of the genes that we're testing are really significantly differentially expressed from the basal to the stimulated state. So there's a huge change here in the transcriptome happening. When we look at what kind of pathways are significantly enriched for these differentially expressed genes, we see terms that we would expect to see. So insulin signaling pathway, response to stimulus, response to hormone, these seem to be weighted towards the muscle fibers. There are also terms that are shared between the different clusters, so cell adhesion and regulation of cell motility. And furthermore, some terms seem to be more specific to the non-muscle fiber types. So even with lower power, we see that the FAPs and endothelial cells may be more enriched for terms like extracellular matrix or vasculature development. And genes in those pathways may be changing specifically in those cell types in response to this stimulus. So that kind of sets the stage for, okay, we've queried how gene expression may be changing across these 4 hours. Now I'm going to change gears a little bit to be talking about genetic regulation, specifically gene expression quantitative trait loci, or these genomic regions. In this case, we'll consider variants, which are associated with differences in gene expression. And on the right-hand side of the slide here, I'm just showing an LZ plot, locus zoom plot, which is a common format that I'll use in subsequent slides, where the x-axis is genomic position and the y-axis represents the strength of association. So points with higher points that are higher in that graph are going to have stronger associations with the expression of a gene. And a similar concept can be done for chromatin accessibility, or CAQTL, where that variant is then associated with either less or greater accessibility of a given region of chromatin. And we can see here that we can do that in many different clusters, and we can represent the accessibility of a chromatin region with a peak, where a higher peak represents greater chromatin accessibility. And when we scan for E and CAQTL across the genome, we detect thousands of significant E and CAQTL that pass our FDR threshold. And one note that I'll just draw attention to here is that if you compare for each cluster, so like type 1 fibers across conditions, pre versus post, you can see that the number of EQTL and CAQTL is roughly on the same order of magnitude from the pre and post context, meaning that there's really this plethora of information that we can derive from both the basal and stimulatory contexts. And that's something that I'll explore further here in the next few slides. And one of the first questions that we asked was, okay, well, do these EQTL in either the basal or stimulatory contexts co-localize with GWAS signals? And by doing so, can they give us hints as to the molecular mechanisms for these nominated risk signals? And this is one example, RS223423, which is a SNP on chromosome 4 that has been nominated as a lead T2D GWAS signal, and it's currently labeled as the SLC9B1 locus, which is a nearby gene. And you can see in the top left there, the GWAS track. And when we do our EQTL scan in the pre or basal condition, we actually see no significant EQTL in our cohort for any nearby genes, including SLC9B1. When we do our EQTL scan in the post or stimulatory context specific scan, we see a significant EQTL for a gene called MAMBA. And the lead EQTL here is in high linkage disequilibrium with the lead T2D GWAS signal. And MAMBA is actually a lysosomal enzyme that plays an important role in oligosaccharide degradation, has recently been linked to type 2 diabetes. And this really drives home the point as to, okay, well, if we're not seeing a lot in the basal context, really understanding these EQTL in a disease-relevant stimulatory context can provide additional and perhaps new insights into what these risk signals or how these risk signals are acting and exerting influence on a disease. So previously I showed you the results here for type 1 fibers, which is in one cluster. But MAMBA actually has this response EQTL, which is an EQTL that is not present in one context, the pre-context, but is present once we look in the appropriate stimulated context. And we can appreciate here that this response EQTL is present across all 5 of the most abundant cell clusters, suggesting that this effect is prevalent across different clusters in skeletal muscle. Now, that was for GWAS signals, but there are also response EQTL for other genes that are not tied to GWAS signals. And because we're using an insulin stimulation, we can ask the question, okay, well, can understanding EQTL in different contexts give us insights into these genes, and particularly genes that may be associated with the insulin signaling pathway. So does the regulation of these genes change across contexts? And I'll show you an example here for a gene, a well-known gene KRAS, which is associated or tied to many things, including cell growth. And as you can see here, it's a central hub or node in the insulin signaling pathway. I'll show results from a few different studies here. So this is from here on this track. I'm showing the TopMed study. So a very large study, including greater than 6,000 samples, and this was done in whole blood and basal contexts. This analysis was led by Dr. Peter Orchard from our lab. And you can see here for KRAS, there's not really a significant EQTL that's emerging for this gene when we look in whole blood and basal contexts. Oops, sorry. When we look at a different cell type, so this is now in skeletal muscle, and this is from the fusion study. This was a study that included almost 300 samples in its EQTL analysis, and this is for skeletal muscle and basal contexts. And again, we see a relatively flat EQTL LZ plot. So there's not really a variant close by to KRAS that we can detect is associated with differential expression in KRAS. When we look at our study, which is also in skeletal muscle, and when we look at the pre-context, this is in basal context. Now, our study only includes 49 samples, but we can appreciate that, again, we see a relatively flat EQTL track. So there's not really a variant that stands out there, which is also really encouraging because that matches with what we see in a much larger study in the fusion study, which was led by Dr. Rishi Varshney. And you may be able to tell where I'm going with this, but so now when we look at the skeletal muscle in a post-clamp disease-relevant stimulated context, again, in a sample size of 49, we can now see that there is a variant that is associated with KRAS expression, and we can appreciate this variant only in the context of skeletal muscle and stimulated context. So even with a sample that is relatively low compared to some of these other studies, context matters for detecting EQTL, and understanding the genetic regulation for these different contexts may give us novel insights. So that was KRAS for our study. And again, KRAS is a gene that has a response EQTL that is seen in all 5 of these most abundant clusters. I'll show one more example from the insulin signaling pathway, and this is for a gene IGF-1. And IGF-1 here for type 1 fibers, you can see here the gene track for IGF-1, and it has an EQTL in the pre-context, but it does not have an EQTL in the post-context. So this regulatory signature is really responding to insulin stimulus, but in the opposite direction. It's quote unquote disappearing, but we still term this a response EQTL. And furthermore, this response EQTL is actually specific to certain cell clusters. So the type 1 fibers and the FAPs, despite really vast differences in power, if you remember. So there are a lot more type 1 fiber nuclei than we have FAPs. And this really highlights the power of that single nucleus approach, coupled with the paired sample biopsies. So to add kind of one more layer to this, we have if we consider a variant that has a response EQTL that is also linked with a GWAS signal, that variant may also be associated with chromatin accessibility. So what about that variant is also a CAQTL? And I'll show a biological model of this. And this is where we can think of the idea of primed enhancers. So in primed enhancers, I'm showing 2 contexts here. Context 1, or the basal context here on the top, and context 2, the stimulated context on the bottom. And you can appreciate how allele 2 is always associated with greater chromatin accessibility. But in the basal context, perhaps TF1 is bound there. It might be a pioneer TF, but we don't really see differences in a target gene's expression. So we don't appreciate an EQTL. But only in a specified context, again, this is a model, perhaps a context-specific transcription factor here, TF2, may come in, bind to this regulatory region, and then the target gene's expression is increased, and we can appreciate both an EQTL, but also have a new way of understanding the regulation for this target gene. And this is an example of a primed enhancer element that we see in our data for TBX15, which is a transcription factor. You can see that there is this chromatin accessibility or the CAQTL signal that is present in FAPs, endothelial cells, as well as many other cell types in skeletal muscle. But there's only an EQTL, and I'll focus here on the type 2A intermediate fibers. There's only an EQTL or a stronger EQTL here in the post-clamp track. So it's a CAQTL under both basal and stimulated conditions, but only an EQTL in that disease-relevant stimulated context. So this could be a primed enhancer element for TBX15. And what's really interesting about TBX15 is it's been implicated in glucose metabolism and muscle differentiation, but specifically in these fast twitch and intermediate muscle fibers. So the mechanism of that is still yet to be deciphered, and analyses like this can give us some more testable hypotheses as to how this gene is being regulated under both basal and stimulated contexts. So with that, I'll wrap up and kind of just summarize. We included 47 individuals with 2 paired samples each, so 94 paired samples from 2 different contexts. We used genetic multiplexing with Multi-Omics to identify 157,000 past QC nuclei. And based on those PCA plots, we can appreciate how the gene expression modality may be more dynamic than chromatin accessibility in response to this stimulation. And some E and CAQTL, as well as primed enhancers, are only detectable in specific contexts. And that really spotlights the importance of context in understanding how disease risk is genetically encoded. In the future and our current work, we're really exploring single-cell EQTL mappers, notably SAGE-QTL, and we really want to functionally validate some of these hits with massively parallel reporter assays and other functional assays, specifically for follow-up of GWAS-linked response EQTL and those primed enhancer elements. So we're really excited about that. And with that, I will say thank you so much to a wonderful team, especially some of my immediate mentors, Adelaide Tovar, Arushi Varshni, and Steve Parker. Thank you, and thank you all for your attention. I'm happy to take any questions.
All right, thanks, Ben, for a really exciting talk. Now we have time for a few questions if you want to type them in the chat and I can read them out. I also have lots of questions. I meet with Ben twice a month, so I could ask him as many questions as I want. So I encourage the audience to come up with some questions. All right, maybe to kick people off, do you want to talk a little bit more about the using SAGE-QTL, using the single cell QTL mapping tools? What do you think? What is the value of using those in addition to or in yeah, other than the normal kind of QTL mapping tools that we've been using?
Yeah, definitely. Thanks, Adelaide. So yeah, we're really excited about this new tool, SAGE-QTL. And let me see, I might have a slide here. Yeah. Oops. Yeah, so SAGE-QTL is this single nucleus approach. And I think, with the approach of our study, it being single nucleus, we're really excited about it boosting power in our QTL results. And in our preliminary results, we do see some good concordance between the pseudobulk technique that we're using. We're using a technique called RASQL right now, and the single nucleus approach. So there's some good concordance there. Still yet to be seen as to which one fully optimized is really going to, give us the most robust results. But yeah, certainly a really exciting technique. SAGE-QTL is also really new, so we've been kind of going back and forth between the developers to kind of, understand the tool and then also hopefully improve it. So we're really excited about some of the coming results from SAGE-QTL. Fingers crossed.
Any other questions? You guys are making it too easy for me. Maybe I'll ask one other question because it kind of came up as you were talking. Have you dug at all into, I know that again, you're still kind of working out the QTL results and getting to a freeze. Have you looked into the location of like cell type specific QTLs versus QTLs that you're seeing shared across different cell types, like whether they're enriched in promoters or enhancers, if they're, overlapping with any of your CQTLs, those kinds of things.
Yeah, that's a really interesting point as to like where the EQTL, if they're shared across different clusters or if they're specific to different clusters. Um, Definitely something that I think we're gonna have to look into. And then also, like you said, enrichment and promoters and enhancers, and then also enrichment for GWAS hits. So do response EQTL that we see across different clusters, are those more or less enriched? For GWAS signals. I think something really exciting to see. But yeah, like you said, we're trying to get to that freeze and really just also trying to tease apart like, oh, okay, well, if we see an EQTL also in REST-QL, but not in SageQTL or something that we see in SageQTL and not in REST-QL, what can that tell us about like the value of the different approach for this sample, but then also perhaps other samples as well. So um, yeah, definitely something that we're going to be working on a lot this week, and then I'll be able to tell you soon.
All right. And then maybe one last quick question from Jacob Kitzman. Any considerations we should think about for using functional assays like MPRA for response EQTLs versus allelic effects generally? So like what types of kind of experimental considerations?
Yeah, yeah, I guess so. For the MPRA, what we're thinking about is like a insulin stimulated glucose response, and then kind of querying the regulatory activity before and after. Um, I can't think of things that I guess you would want to look out for besides, for traditional MPRAs. I think one of the things that will be really important is like variant selection. So, trying to really pick those variants carefully as far as overlap with CAQTLs, overlaps with chromatin accessibility peaks that we see in our data, right? So we have a Hispanic or self-reported Hispanic ancestry. So I think that would be really important. Um, yeah, I think again something that is going to come up in future work. So